Hello boys, welcome back to another session of video tutorials. This session is meant for class 11th biology students and this is meant for 8th August 2020. As we have been discussing about the possible functioning of photosynthesis and the mechanism of photosynthesis, we understand there must be light harvesting centers, and the light harvesting centers are of two: photosystem 1 and photosystem 2, PS1 and PS2, and these two are interlinked through ETS, electron transport chain. So in this, as we discussed in our previous session, two energy molecules, energy compounds are formed. One is ATP, another is NADPH. How exactly this ATP gets synthesized is through a hypothesis. This hypothesis is called chemiosmotic hypothesis. Chemiosmotic. It was propounded by Peter Mitchell in 1961 in the case of mitochondria and chloroplast. He was awarded suitably a Nobel Prize for Chemistry in 1978 for this discovery. This hypothesis explains the relationship between potential energy present within the concentration gradient of ions. Of course, here we will be talking about H plus ions. Remember, during splitting of water, H2O, last session I explained to you, H plus and OH minus will be formed. We were talking about the possible electron movement from PS1, PS2 to electron chain to PS1. So we were making the electron movement and possible movement of electrons resulting in formation of ATP and NADPH. But what about H plus? So that is what we will be discussing about this, that H plus ion concentration. So the concentration gradient of H plus ions across a membrane and the use of potential energy in the form of ATP. Basically, this is to explain about proton gradient and how proton gradient maintaining within the chloroplast, particularly in the lumen, help ATP synthesis. According to this view, electron transport both in respiration and photosynthesis produces a proton gradient, H plus gradient. Here in photosynthesis, the gradient develops inside the thylakoid membrane, particularly lumen in chloroplast. So primary acceptor of electron is located in the outside of a thylakoid membrane, as I will show you here. Look at that. This is what happens. Okay. So you have got a lumen here and you can see H plus ion concentrations. You have got photosystem 2 and photosystem 1 interconnected through electron transport chain, and in electron transport chain many pigments will be there: cytochrome A, cytochrome B, cytochrome F. Okay, and the electrons process through that and results in the formation of NADPH. So primary acceptor of electron is located in the outer side of thylakoid membrane. You can see here. It transfers its electron to H carrier, hydrogen carrier. The carrier removes a proton from matrix while transporting electron to the inner side of the membrane. The proton is released into the lumen, as you are seeing. Proton is all H plus, H plus. H plus is nothing but proton. The proton is released into the lumen while the electron passes to the next carrier, as we are seeing from PS1 to electron transport chain to PS1. Okay, electrons are getting transported. Remember that cyclic and non-cyclic photophosphorylation. Yeah. So the NADP reductase is situated on the outer side of the thylakoid membrane, as you are seeing. NADP, it is reductase. So it obtains electrons from PS1 and protons from matrix. To reduce NADP, as you are seeing, NADP plus to NADP plus H plus. The consequence of these events is that concentration of protons decreases in matrix or stroma region, while their concentration in thylakoid lumen rises. So there are two areas with a difference in H plus ion concentration, resulting in decrease in pH. So the influence of H plus will result in difference in pH. So a proton gradient develops across the thylakoid membrane. The proton gradient is broken down due to the movement of protons through transmembrane channels. So you got the ATPase, that is, you can see F0 and F1 particles. So it is the F0, F1 will be on the stroma and granum, on the granum side of it. So as protons move to the other side of ATPase, as you are seeing, they bring about the conformational changes in F1 particle of ATPase, or it is also called coupling factor. The transient F1 particle of ATPase enzyme form ATP from, as I explained yesterday, ADP plus inorganic phosphate. So ADP plus inorganic phosphate will form ATP. So as you are saying, ADP, of course, the ATP synthase enzyme which helps in formation of ATP will make the inorganic phosphate present in the lumen to combine. As a result, the one molecule of ATP is formed when 3 H plus are passed through the ATP synthetase complex. So in order to form the ATP, how many H plus are required to pass through the complex? At least 3 H plus should be there. So this is how the chem-osmotic hypothesis proposes that as long as proton gradient is maintained within the lumen, it results in the formation of two energy compounds, ATP and NADPH. So there is the same thing we are going to write here. The chem-osmotic hypothesis has been put forward to explain the mechanism of ATP synthesis, which is linked to development of a proton gradient H plus, okay, across membranes of the thylakoid. Here the proton accumulation takes place towards the lumen. The protons that are produced by splitting of water are accumulated inside the lumen of thylakoid. As the electron moves through the photosystems, like photosystem 1, 2, protons are transported across the membrane. NADP reductase enzyme is located on the stroma. You remember in a thylakoid there are two areas, grana and stroma. So it is located in the stroma side of the membrane along with electrons from the acceptor. It removes H plus from the stroma during reduction of NADPH plus H plus. This creates a proton gradient across the thylakoid membrane as well as measurable decrease in the pH in the lumen. ATP channel that allows diffusion of protons back to stroma across the membrane. This releases energy to activate ATPase enzyme that catalyzes the formation of ATP. That means in the formation of ATP, what is most important is activation of an enzyme called ATPase. ATPase gets activated only when there is pH, decreased pH. And the decreased pH will be possible only when there is a proton gradient. So chem-osmosis requires a membrane, okay, and a proton pump, a proton gradient and ATPase. Energy is used to pump protons across the membrane to create a gradient or a high concentration of protons within thylakoid lumen. And how this is used? This ATP, NADPH and O2, all these are byproducts of the light reaction. So O2 diffuse out, we are thankful to plants, we are taking it of the chloroplast, while ATP and NADPH are used to drive the process leading to the synthesis of biosynthetic phase, that is starch formation. This process does not directly depend on the presence of light. It does not require any light, but is dependent on the products of the light reaction. That's how both are light reaction and dark reaction are interconnected. So ATP and NADPH besides CO2 and H2O should be there. So there is one great scientist, Melvin Kelvin, after World War II, worked with radioactive C14 and studied algal photosynthesis, which led to the discovery that the first CO2 fixation product was a 3-carbon organic acid. It is called PGA. 3-PGA. PGA stands for phosphoglyceric acid. And how this